friends to a new session on genomics and proteomics. In this session, we will discuss family studies and complex traits genetic maps. Gene mapping is the process of finding the location of genes on each chromosome. It is an important step in identifying the genes involved in a genetic disease. Once a disease gene is accurately located, we can determine its DNA sequence and its protein product. For example, cystic fibrosis is the most common lethal inherited disease in the United States. As many as 1 in 2,500 Americans of Northern European descent carry a gene with CF. In 1985, the gene was mapped to chromosome 7q31q32 by linkage analysis. 4 years later, Francis Collin and colleagues determined its DNA sequence. Now we know that the disease is caused by the defect of a chloride channel encoded by the CFTR gene. Relative to linkage analysis and sequencing, genome-wide association studies have less power in cases where different rare mutations act in different families or individuals at the same locus, that is allelic heterogeneity. However, they are far more sensitive than family studies to complex polygenic associations in which a phenotype is associated with the joint effect of many weakly contributing variants across different loci, that is locus heterogeneity. In this sense, GWAS have been a resounding success, identifying thousands of disease associated loci and revealing previously unknown mechanisms for conditions such as Crohn's disease, type 2 diabetes and macular degeneration. Family studies. Family studies have been used in the genetics since its beginning. One of the most successful approaches to identify new genes responsible for a disease has been the search of the full genome in families of affected individuals. Mendelian ratios were based on a relationship between sweet peas. The offspring get their genetic information from the parents, so they can have different genes than them. That is a principle of family studies. Pedigree, also called family tree, is a graphic parallel to a family relationships. Family studies have been used to identify monogenetic diseases, highly penetrant traits of a biomedical importance, including a variety of inborn errors of metabolism and their genetic diseases. Example, cystic fibrosis, Duchenne muscular dystrophy and Huntington disease. The importance of family studies. Family studies have several favorable features for gene discovery. Studies of extended pedigrees or even nuclear families are likely to represent a more homogeneous and limited set of causative genes and pathways. Members of the family usually share not only genes but also the environment. So, if only one or more but not all members of the family are affected, we can exclude the external influences and recognize the origin of hereditary diseases. The special twin study case is a perfect example of how the environment influences the origin of the disease. On the other hand, distances is the major disadvantages of family studies. Sometimes it is more expensive to create a pedigree of affected families than the unrelated people because the relatives can live far away. There are some typical diseases which are connected with higher familiar occurrence: schizophrenia, bipolar disorder, suicides or eating disorders. The use of family trees. Genetic linkage, genetic mapping, finding the origin of disease.